So good morning. So I'm going to be talking today about the use of CRISPR-Cas9.
So let's go to adjust the microphone.
Is that better?
Yep.
Yes.
So I'm going to be talking today about the use of CRISPR-Cas9 genetic modification technology in humans. So this is already happening at the moment, so it's not just a theoretical possibility anymore. So we're going to talk a bit about what's going on at the moment in this field, what's about to happen in the near future, and then I'll talk a bit about some of the ethical and philosophical implications of this work. So first I figured I'd tell you guys a bit about myself and what I do for a living. So I'm a scientist who works at the University of Auckland. And I first got interested in CRISPR-Cas9 technology when I started my PhD project. So my PhD was on Alzheimer's disease. And it was widely acknowledged when I started the project that the biggest impediment to work in Alzheimer's research was the lack of a suitable animal model. So lots of mouse models and small animal models of Alzheimer's disease have been developed using a whole variety of different genetic modification techniques. But ultimately they didn't live long enough and they were too genetically different to humans, so we were never able to create a model that captured all of the symptoms of Alzheimer's disease. Obviously the best model for a human disorder would be a primate, but unfortunately they're incredibly difficult and expensive to work with, and there's also a lot of ethical issues. So increasingly, and particularly for neurological conditions, farm animals are being considered a good trade-off between the limitation of a rodent and the difficulties of working with a primate. So the purpose of my PhD project was to create a sheep model of Alzheimer's disease. And in order to do that, I had to genetically modify the sheep. And this was so that we could capture the pre-symptomatic phase of Alzheimer's disease at an earlier age. So in humans, we develop Alzheimer's disease about 10 to 30 years before any symptoms become apparent. So nowadays that's considered to be the best kind of treatment window, because if you can fix the disease at that point, it'll be before any symptoms occur, and before a lot of brain damage has occurred as well. So we decided to use CRISPR-Cas9 technology to make these sheep. And back then it was still quite new, since this was 2016. So we didn't really have a clue what we were doing with it. We were just trying different things, and I was closely following the research as all this was developing and trying to incorporate things that work for other people into my own research. So eventually I was able to create a handful of sheep that had the genetic modification that we wanted, finished my PhD at that point, and now I'm still working on the same project, but now I'm tracking these animals as they age, collecting data on them, and trying to prove that they will indeed be a good model of Alzheimer's disease. And once I do that, pharmaceutical companies will probably want to use them to test therapies. And alongside that work, I'm also doing some work developing CRISPR-Cas9 systems that can knock out mutations in genes that cause Alzheimer's disease, with the hope that they'll one day be used to prevent Alzheimer's. Cool. So this presentation is broken into 3 parts. So the first part will be, I'll talk about what genetic modification is and why CRISPR-Cas9 was such a revolution in this field. Most of the really interesting stuff happening right now with CRISPR-Cas9 is in medicine, so I'll talk about that. And then lastly I'll talk about the much more controversial topic of using CRISPR-Cas9 technology to genetically modify human embryos. So the original version of genetic modification technology was called transgenesis. So this is basically where a gene was taken from a foreign organism and then put into a host cell. So one really famous example of this, as you can see in that picture there, some scientists took a gene from a jellyfish and they put them into mouse embryos to create mice that will glow green if you shine a blue or a UV light on them. And so this technology was used to do all sorts of wonderful things, but it always came with a series of downsides and limitations. And one of them is the fact that these transgenes tend to integrate into the host genome at random. So it's very hard to control where they end up, and occasionally they'll end up in the middle of native genes, and that will usually ruin their function. It was also very hard to control how many genes inserted. So this technology basically works by firing hundreds of copies of the transgene into the cell and then hoping that at least one of them sticks. But sometimes dozens of copies would end up inserting all in different places. Some of them would only partially insert and not be functional. Some of them would end up inserting completely, but they'll be back to front. So I think the end result was often a bit of a mess. And the other problem, particularly for modelling human diseases in an animal, is the fact that these genes, so when you put a gene into a cell, you need to add regulatory elements to tell the cell what to do with it. And for a variety of technical reasons, these regulatory elements were almost never the ones that came with that gene in the organism that it was found in, and it was also not the kind of regulatory elements that would be used in the host cell for a gene of that nature either. Which meant often these genes would be active in the wrong organs, or sometimes they're only meant to be active in certain phases of development, but they'd end up being active the entire time, which will make it quite difficult to separate the effects of a disease-causing mutation from all the artefacts of transgenesis. So all these problems can be solved with another technology called targeted gene editing. So this is where, instead of introducing foreign DNA, you create little mutations in the native DNA of a cell. These mutations tend to be a lot smaller than with transgenesis, and they also tend to be much more precise. And because we have 2 copies of each chromosome, and 2 copies of every gene, if you target this technology to a location, there's only up to two mutations you can end up with. And also for creating animal models of diseases, this is a lot better because it means that you put a mutation into a native gene, it's sitting next to all its natural regulatory elements, so it behaves like a normal gene. So transgenesis technology has been in use since about the mid-70s, and targeted gene editing has been around since about that long as well, but the original version of this technology was highly inefficient. So it was almost never used outside of certain work that was being done in mice. Along the way, people developed ways to make it more efficient, but then usually these methods came with limited specificity, which meant that this technology could only be targeted to certain places in the genome, but not others. And it was also relatively complicated to program, so it was difficult to tell this particular system where to go in the genome. Pretty much all of that was solved with CRISPR-Cas9. So CRISPR-Cas9 is a highly flexible system that can be targeted almost anywhere in the genome. And it's also very cheap and easy to use. So very quickly it was adopted by pretty much every genetics lab, and now we use it to do a whole variety of different things. Cool. So CRISPR-Cas9 was discovered and developed by 2 scientists who first published their work in 2012, and then they later went on to win the Nobel Prize in Chemistry in 2020. And this system initially comes from bacteria, so it's part of the bacterial adaptive immune system. It's quite a clever system, so when the bacteria gets infected with a virus, it will take little bits of viral DNA and then insert them into its own DNA next to CRISPR elements. And then it will make copies of those little bits of viral DNA attached to the CRISPR elements, and they float around in the cell looking for a match. And if the bacteria gets infected by the same virus again, the Cas9 enzyme will chop the viral DNA in half, and that renders it inactive. So I'll just talk quickly about how CRISPR-Cas9 works for us in the lab. If you don't understand this part, it doesn't really matter for the rest of the presentation. So this here is a representation of a DNA strand or a chromosome, and it's been pulled apart in the middle here. So this grey blob is the Cas9 enzyme, so this is what actually does the work. And this pink thing is what we call a guide. So half of the guide is designed to be complementary to the target sequence, so this is how it's targeted. And then the other half is designed to bind to the Cas9 enzyme. So when we put these into a cell, the Cas9 enzyme and the guide will whiz around the genome looking for a match. When it finds a match, it'll cut the chromosome in half at a specified location relative to the target sequence. So that results in a broken chromosome. The cells really don't like having broken chromosomes, so they very quickly will activate DNA repair pathways in order to fix the break. And the most common one they activate is basically just an emergency repair pathway that will grab the two ends and stick them back together as fast as possible. It's highly error prone, so often it will accidentally delete a few bases in the process, or it will insert a few that weren't meant to be there. So you end up with a little interruption in the chromosome, and if that's in the middle of a gene, it will interrupt the coding sequence and switch the gene off. And often that's all we have to do. So if we want to study a gene, for example, the first thing we'd do is to switch it off in a cell line and just see what happens. But there's also a lot of diseases that can be cured this way if you just switch off the gene that has a mutation in it, or if you find a gene in a relevant pathway where switching it off will block the effect of a disease-causing mutation. Cool. There's a second DNA repair pathway we can take advantage of. This one's a bit more difficult because it's only active in certain phases of the cell cycle. But this one's much more accurate. So this is where the cell it'll find the two ends of the broken chromosome, and then it will look for the matching chromosome because there's always two chromosomes in the cell. So it will find the intact chromosome, bring over the broken chromosome, and then it will use the intact chromosome as a template, so it copies over the sequence across the break site to make sure that it gets repaired accurately. And we can take advantage of this by introducing lots of bits of artificial DNA that are quite short, but almost completely match the bit where we're creating the break in the chromosome, and then we put, say, like a one base pair mutation in there, and then hope that the cell mistakes one of these artificial bits of DNA for the other chromosome, and if it does, it will use them as a template and it will copy over our mutation of interest. And this is quite handy because it means that we can swap nucleotides, and if we can also create insertions and deletions that are a specific length. So this is a very precise way of changing things. So by far the most consequential application of this technology at the moment is in the field of medicine. So the first important thing to understand about this is that these genetic modifications are only being made in somatic tissue. And somatic tissue is basically just all non-reproductive tissue. So if you genetically modify somatic tissue in an individual, they won't be able to pass that on to any offspring that they have, so it stays with the individual. And there's an awful lot of disorders that could potentially be cured this way. So for example, there's about 6,000 single gene disorders that could potentially be fixed. But there's also a lot of multi-genetic or non-genetic disorders that could potentially be targeted as well if scientists can figure out how to modify a gene in the relevant pathway in order to block the disease or the mutation that's causing a disease. And probably the biggest benefit of using genetic modification to treat diseases is that it will typically just be a one-off treatment that will then fix the condition at its source. And there has already been an attempt to do this. So some of you might remember the attempt to genetically modify some children to treat a condition called bubble boy disease back in the 2000s. So this was a severe immune condition. And it did actually work. So all 20 children that were treated this way were cured of their immune disease. But unfortunately 5 of them later went on to develop leukemia. So this was with the old version of genetic modification technology. This was with transgenesis. So it was later determined that in those 5 children, one of those transgenes landed in the middle of a cancer-suppressing gene. So after that, this work was halted. And pretty much for about 10 years, nobody was really talking about using this to cure human diseases. But research was still going on in the background. And it wasn't until the advent of CRISPR-Cas9 that this really began to look like a real possibility again. So clinical trials using CRISPR-Cas9 therapies began about 5 years ago. And the first CRISPR-Cas9 therapy to enter clinical trials was trying to treat sickle cell disease and related conditions. So this is a genetic disorder that's caused by 2 faulty copies of the beta-globin gene. The beta-globin gene codes for the beta-globin protein, which is an essential component of hemoglobin. And hemoglobin is what transports oxygen around the body in red blood cells. So having 2 mutated copies of beta-globin creates deformed blood cells that don't move through the body properly, and they tend to cause blockages. It causes a range of symptoms, including severe anemia, so-called pain crises, and strokes. And the treatments available were only just kind of moderating some of the symptoms, so they included regular blood transfusions and pain management. So when we are a fetus, we don't use beta-globin at all, so we use another version called gamma-globin, coded for by a different gene. And this gene is later inactivated by a repressor gene, which switches it off, and after that we become completely reliant on beta-globin. So this happens over the first year of life, and it correlates with the appearance of disease symptoms in those who have sickle cell disease. But some scientists discovered at some point that people who have a natural mutation in their repressor gene continue producing fetal gamma globin for their entire life, but where they also happen to have beta globin mutations, they have much milder forms of sickle cell disease. Cool. So some other scientists decided to take advantage of this fact and try to create a CRISPR-Cas9 therapy that could be used to inactivate the repressor gene, so in effect leaving fetal gamma globin on throughout the lifespan. So they did this in some patients by extracting bone marrow stem cells, genetically modifying them in the lab, and then replacing them to the patient's bodies. And those clinical trials were started in 2019. They turned out to be very successful in reducing symptoms, so they almost completely removed major symptoms from the patients. And over the time span of the clinical trials, no serious side effects were noted. So then this therapy went on to become the first CRISPR-Cas9 therapy that received FDA and other regulatory approval, and that only happened in December of last year. Cool. So the next disease in the pipeline, so this is CRISPR-Cas9 therapy designed to target a disease called hereditary transthyretin amyloidosis. So you probably won't have heard of this one. It's a fatal heart disease controlled by the liver. So it's caused by a faulty transthyretin gene that then produces a faulty transthyretin protein. But although this protein is produced in the liver, it gets released into the bloodstream where it then travels around and lodges itself in the walls of the heart. But it's usually fatal within a decade of diagnosis. So for this particular treatment, CRISPR-Cas9 was injected directly into the bloodstream of the patients. Normally this wouldn't work if you're trying to target a particular organ. It would be a very inefficient way of trying to get the reagents into the organ. But fortunately, the liver loves to suck things up out of the blood. That's part of its job. So in animal studies, most of these CRISPR-Cas9 reagents and most of the genetic modification was occurring in the liver. And this therapy is designed to knock out both the normal and the mutant copies of the gene, so it just knocks out everything. But in a phase 2 clinical trial, it was able to cause a 95% reduction in symptoms. So it's looking very promising, and at present it's in a phase 3 clinical trial, and we'll have the results from that sometime in 2026. But if this is successful, this will probably be the second treatment that will be approved. So there's an awful lot of other therapies that are currently in clinical trials testing CRISPR-Cas9. So the website CRISPR Medicine News lists 190 different therapies that have been tested. These are still in phase 1 or phase 2, so quite early. but they're targeted to a surprising variety of different conditions. So for example, there's more blood and liver diseases being targeted, along with immune system disorders. In terms of inherited conditions that tend to be quite well known, Duchenne muscular dystrophy and type 1 diabetes. There's also a few looking at heart disease. So these are all targeting a particular gene where when switched off it prevents the production of cholesterol. So it's been tested in people who have genetically high cholesterol, who are at high risk of a heart attack, but also one of them is looking at people who have an acquired level of high cholesterol as well, so this would be a preventative medication. There's over 70 studies that have been targeted to different kinds of cancer. Cancer can actually be targeted in quite a few different ways. So one example is just to try to fix the mutation that caused the cancer in the first place. But you can also try to reprogram immune cells so that they'll attack the cancer cells, and there's a variety of other methods as well. And interestingly, even transmissible diseases have been targeted with CRISPR-Cas9 therapies. So a lot of viruses work by lodging themselves in the human genome, so for example the HIV virus. So we can target CRISPR-Cas9 therapies to find the virus in the human genome and put little mutations in it in order to inactivate it. And then there's also some trials going on looking at curing bacterial infections, particularly the ones that cause urinary tract infections. So there's probably many more diseases that could also be targeted with CRISPR-Cas9. Sorry, I've got a bit of a cold. So because CRISPR-Cas9 is such a flexible system, it can be targeted to almost any gene, and there's an almost unknown number of different genes or genetic pathways we could modify in order to tweak certain things and potentially prevent different diseases. So I expect in the next several years there'll be a lot more things entering clinical trials, and if anyone's interested in what's being treated, what conditions are being looked at, I'd recommend going to the website CRISPR Medicine News because they collate all the information from the different clinical trials that are going on at the moment. Cool. So despite the incredible promise of CRISPR-Cas9, there is still the potential for some problems. The greatest one is still the risk of off-target effects. So this is drastically less likely than with the previous generation of genetic modification technology, but it does sometimes still happen. Some of this risk can be mitigated by doing studies beforehand in cell culture or in animals, figuring out which locations are likely to be a problem in this regard. But the biggest concern is always going to be getting a mutation in a cancer-suppressing gene, because this will lead to uncontrolled cell growth. And the reason this is so concerning is because you only need to get one mutation in one gene to cause a serious problem. Another problem inherent to this technology is unlikely, but you can sometimes get chromosome rearrangements. And that tends to happen if the Cas9 enzyme accidentally cuts two different chromosomes at the same time. So if a cell activates the emergency repair pathway, it will just grab two ends and stick them back together, but they won't necessarily be the right two ends. And so this can then separate genes from their regulatory elements and cause downstream effects. In very extreme cases, you can get shattering of a chromosome, although that is very unlikely. Another issue is that it's very difficult to assess the outcome. So at present, when we genetically modify a cell and then want to find out exactly what we've done to it, we have to kill the cell in order to test the DNA. Which means that we can't genetically test every cell that we genetically modify or will destroy the intended outcome. Which means that it'll probably be decades before we know the full extent of any problems that are going to come up. Which is why at present this technology is limited to people who have either very severe conditions or lethal conditions where the potential benefits are likely to outweigh any possible risk. Another potential issue, so this is unlikely, but there is the potential risk of modifying reproductive cells if you inject CRISPR-Cas9 directly into the bloodstream. So this isn't really something that anyone worries about when cells are taken out of the body and then genetically modified in the lab, as with sickle cell disease, because CRISPR-Cas9 has a very short lifespan. Like, it'll get degraded by the cell pretty quickly and it won't last more than a couple of days. So all you need to do to mitigate that risk is just have a long enough wait time before you reintroduce the cells. But if you if you inject it directly into the body, even though probably most of it will just end up in the liver, there is the chance that some of it will end up in other organs and tissues as well. And I know it might sound like a good thing if you fix a mutation that causes a disease with genetic modification and then pass that on to your offspring. But it does really depend on how the disease in question is being fixed. So at present, it's still a lot easier to switch genes off than it is to precisely change them. So a lot of the therapies are basically just finding a gene that can be switched off in a pathway that then blocks the effect of a mutation. Sometimes it'll be the gene that's mutated itself, sometimes it will be another gene. And some of these genes are really not that important when we're an adult, so often they don't have much of a function, so they don't cause any side effects when they're switched off. But at least some of these genes will be active at different phases of development, so you don't really want to put a mutation in one of them and then pass them on to your offspring, or there's a potential risk of developmental defects. So along with the potential biological risks, there's also the potential for misuse of this technology. And this really came to light with an individual called Josiah Zania. So this is a guy who got a PhD in biophysics and is now a self-identified biohacker. He apparently found academia too hierarchical and decided he was going to bring CRISPR-Cas9 to the people. So he originally started by testing CRISPR-Cas9 therapies on himself. So he was trying to knock out his myostatin gene to enhance muscle growth. And apparently at another point he was trying to change his skin colour. He then started selling home CRISPR kits to the general public along with instructional videos. And so this, along with some other stuff he was doing, got him into trouble with the FDA and a lot of other regulatory bodies. He was featured in a documentary called Unnatural Selection. His argument is basically that people have the right to do what they want with their own bodies. I'm sure there is some philosophical argument there. But it did bring up a lot of issues around, for example, informed consent. So because of his background, he probably does understand the risks, but it's not clear that anyone who was copying his methods would necessarily understand what they were getting themselves into. So far there's no evidence anyone's been successful at trying to genetically enhance themselves in this way, but the technology to do so will probably be with us in the not too distant future. So it does raise the question around how, as a society, we want to be limiting and regulating this technology. So for a lot of diseases, it would be a lot easier to fix them at the embryo stage than to try to fix them in an adult once the disease is already developed. So in this case, genetic modification would be found in every cell of the body and would effectively cure the disease before it ever has a chance to develop. However, these modifications will also be in reproductive tissue and will be potentially passed on to any offspring that that individual has. So for couples who have severe hereditary conditions who want to avoid passing them on, we do have one technique available already. It's referred to as negative selection. So this is where embryos are created via IVF, and then pre-implantation testing is done on the embryos to determine which ones carry the mutation, and then only the healthy ones are implanted. However, this doesn't work for every genetic condition. So for example, where both parents have what's known as a homozygous recessive condition, so this is where you need 2 faulty copies of the gene to have the disorder, and if you have 2 parents with the same condition, they've got 4 faulty copies of the gene between them and no healthy copies. It can also be a problem if one parent has 2 copies of a dominant mutation. So dominant mutations are where you just need one faulty gene to cause the problem. If one parent has 2 copies of this, then all of the children are going to have at least one. And there also does seem to be an argument that it's somehow wrong or potentially immoral to discard embryos that have mutations, and it would be better if we could just fix them instead. When consulted, families with these kinds of inherited disorders do really seem to want this technology to be available, particularly when they don't have other options. So in this case, they're either stuck with the option of not reproducing or having children that have a disorder. But it does raise a lot of ethical and philosophical questions. So the strongest argument against this is the fact that it's impossible to cure someone who doesn't exist yet, which means that this would be more about the needs of the parents than the needs of the child. And it raises the question of whether people have a right to biological offspring as well. So in terms of technicalities, there's no reason we couldn't do this in human embryos. So we've been modifying the embryos of all sorts of other species for a long time now, and human embryos are no different from a biological point of view. And scientists were doing work trying to genetically modify human embryos in the lab, they just weren't implanting them. And that was mostly just to test different therapies and to see kind of where our technology is at. That changed in 2018 when a Chinese scientist called Dr. He announced at a conference that he had already genetically modified 2 children. So the gene he modified was the CCR5 gene. So this gene produces the CCR5 receptor protein that sits on the outside of cells and is what the HIV virus uses to bind to the cell and gain entry into the cell. So people who naturally have mutations in their CCR5 gene, where they have both copies mutated, they don't produce a CCR5 receptor and are therefore immune to HIV. So this is not very common, it's only found in about 1% of the Northern European population. But the child's father in this case was HIV positive, so that was the reason that the scientist gave for doing this experiment. He presented this as a positive thing for humanity, but instead everybody just panicked. So he was arrested, sacked by his university, and then spent 3 years in prison. And I think part of the reason for this was that there was no explicit laws against doing this, but all of the work that gets done in humans or animals has to go through an ethics committee and they have to sign off on it, and I think it was just widely assumed that no ethics committee would sign off on this work at present. So as a result of this, we now have much better laws and regulations in place telling people what they can and can't do. So it's still not entirely clear what the result of this experiment was. So there turned out to be 3 genetically modified children, all of them girls. He only presented data on the first 2 at the conference. And a few experts who were there at the conference later said they thought the children were probably going to be mosaic based on the data that the scientists gave from pre-implantation testing in the embryos and then genetically testing the girls once they were born. So mosaicism is where you have the modification in some cells but not others. And I'll talk about that on the next slide. But basically it's unclear at present whether these children have HIV immunity or not. There's also other ways to stop HIV transmission if a father is positive, so this was deemed medically unnecessary. However, as a proof of principle experiment, this was actually a really good gene to be targeting, so it's now widely assumed that that was what he was doing. However, there was some controversy around the use of this particular gene. So even though it prevents HIV and appears to prevent other conditions as well, essentially by removing the CCR5 receptor, a tiny piece of the immune system has been deleted, and it does appear to make people more susceptible, at least to one other virus that we know about. So the death rate in people who lack the CCR5 receptor is much higher if they're infected with a virus called the West Nile River virus. We don't really know about every condition in this particular variant, so this is kind of one of those things where it might be really good in some environments and really bad in others. And then the other controversy that broke out a bit later is over the fact that this particular gene has been studied in 2 parallel lines of research. So one is HIV and immune research, and the other is intelligence research. So if you knock out both copies of this gene in a mouse, the mice automatically become 1.5 times smarter, and they also appear to have quite significant memory enhancements. It's unclear how this translates to humans because we just don't have a lot of data. There's not many people that lack both copies of this particular receptor. And mice brains also are not necessarily the best models of human brains, as I found out doing my sheep research. So we really don't know whether the scientist has affected the brain development of these baby girls or not. Well, so by far the biggest technical problem with modifying embryos is the issue of mosaicism. So this happens because the CRISPR-Cas9 reagents get degraded quite quickly, like within a couple of days. But when you create an embryo, as soon as the egg and the sperm meet to create a single-celled embryo, things happen really quickly. So very quickly that one cell will split into two cells, which then splits into 4 and then into 8. And the CRISPR-Cas9 reagents do tend to linger through subsequent cell divisions. In my own work with sheep, I found in one case they lingered up to the 8-cell stage. That wasn't very common, but it is quite typical to see them hanging around up to the 4-cell stage. So sometimes only a small proportion of the organism has the desired modification, and whether that's a problem or not really depends what kind of condition you're trying to treat. The other thing is that when you have a single cell, there's only 2 copies of the gene that can be modified, so at most you can get 2 different mutations. However, once that cell splits, particularly by the 4-cell stage, you now have 8 copies of the gene, which means that you can potentially get up to 8 different mutations in that one organism. And in my work I did with sheep, there were a couple of times where we had lambs born that had 5 different mutations, simply for this reason. Now, whether that's a problem or not, again, depends what you're trying to do. A lot of work has been put into developing technologies to try and remove this problem. So all of them have lowered the rate of mosaicism, but none of them have been successful enough, but none of them have gotten anywhere near like 100% removal of this problem. So kind of at this point it's considered that it's too risky to be doing this in humans when we don't really know what we're going to create. Like we can't guarantee we're going to modify that embryo at the single cell stage. And the other issue is that pre-implantation testing is really unreliable when you're testing mosaic embryos. This was the bane of my own research that I was doing in sheep. So if you inherit a mutation, it's found in every cell of your body, which means that if you have an embryo, all you have to do is pull off a few cells and test them, and that will tell you what's going on in the entire embryo. However, if the embryo is mosaic, you have no idea whether those few cells you just pulled off are representative of the entire embryo, or whether you've just pulled off the few cells that did actually get a genetic modification. So if you're doing this in animal research, it just means that you have to test a lot more embryos, implant the ones you think have the mutation, and then just wait to see what happens when the animals are born, because then you can get an accurate readout of what happened. So this might be a bit of a pain for animal research, but we can get around it. However, no ethics committee is going to sign off on creating a dozen human babies and then selecting the 2 or 3 that you want and discarding the rest. So we really need to be sure what we're doing, I think, before we start implanting genetically modified embryos. There's a lot of attempts to overcome these problems at the moment, and given the pace of technology development in this field, I would expect probably within the next 5 to 10 years we would have figured out how to deal with this issue, possibly even sooner. So there's a general sense in the scientists who do this work in human embryos, the ones that are not implanted, that probably this will be happening in the future, in which case there's some questions we really do need to think about. One of them is how many disorders should we be removing, and another one is which kind of disorders we should be removing. So for example, it's quite easy to argue that we should be removing mutations that are going to be lethal in childhood, for example. Like, it's pretty hard to argue we should keep those. But there's an awful lot of mild conditions out there that could potentially be treated with gene therapy, but we do want to have a think about whether we want to do that and how much we would reduce the diversity of the human gene pool if we just start removing everything that's associated with a problem. Other issues are that some genes really have environmentally determined outcomes. So this is like the CCR5 gene that was altered in those baby girls. Might be really good in some environments and really bad in others, and then you've got to try and guess which environments those people are going to live their lives in. This is kind of similar to the problem, I think, with the drug Ozempic, where people are arguing, like, should we just solve the environmental problem of obesity rather than giving everyone a drug to keep them skinny? There's also other conditions that we describe in science as not being fully penetrant, which means that only some people with that mutation are going to get the disorder. It usually depends on the other gene variants that person carries or the environment. And probably the one that I worry about the most from a scientific point of view is that a lot of genes have multiple roles. So they often do different things in different organs, and they can also play different roles at different stages of development. There's about 20,000 genes in the human genome, and we really don't know what half of them are doing all of the time. So there is the possibility that you're going to interfere with things you never intended to interfere with, and I think, just from my own point of view, we really need to have a better understanding of what these genes are doing before we start messing with them. And then the other issue that tends to be quite controversial is gene variants that cause things like autism and neurodiversity. So these mutations and variants can sometimes confer exceptional abilities, but they can also sometimes confer disability. So it isn't really clear whether we should be removing these from the population or not. And the most controversial thing is the idea of positive selection. So this is the opposite of negative selection, where you're trying to remove variants that cause diseases. This is selecting for positive traits, so trying to enhance people, basically. One example, potentially, is the CCR5 gene that's already been modified in those girls that potentially might enhance their intelligence, and we just don't know. But there's also other genes implicated in intelligence that could be targeted in this way. And then there's, of course, all sorts of other things you could think of that people might find desirable in their children, enhancing athletic performance or attractiveness. It depends, this could also even become a fashionable thing. If people want, more children with blonde hair or something, there's a lot of things you could interfere with to designer babies. And I think it really raises the question of whether it's important to be natural. So there's definitely people who argue in favour of positive selection, and their argument can generally be summed up as, if we can make people better, why not do it? The scientist who genetically modified the baby girls is unapologetic. He only said that he acted too soon. So in summary, CRISPR-Cas9 has created a major revolution in the field of genetic modification technology. It has great promise for curing an awful lot of diseases. I don't think we even really understand how many could potentially be treated at this point. However, there definitely are still some risks, so we'll really have to wait and see over the next couple of decades and just find out whether this is going to be worth doing or not. And we're also getting really close to the possibility of being able to accurately genetically modify human embryos, which could then potentially permanently alter the gene pool of the human species. So I think it's really just a good idea to know what's going on and what the potential implications of this technology is, so that we're able to then discuss and debate how we want to use it. I'd just like to acknowledge my supervisors for my PhD at the University of Auckland, and also one of my master's students who's continuing with some of this work. And I just want to say a quick thanks to Jude, Emma and Chris for encouraging me to give presentations and write articles on this topic.
That was brilliant. A round of applause. I don't think my brain would have been stretched this much if instead of being here I was sitting in a church. So,